All right, welcome back. So we had mentioned those definitions just at the very end of our last video. So let's just take a quick look at them again. So again, sarcolemma is simply going to be this cell membrane. These T-tubules, or these transverse tubules, are tiny invaginations. So they're little areas where the cell membrane is essentially going to dip in. And they quickly spread the action potential to all the parts of the muscle fiber. So as I was mentioning before, as that nerve signal washes along that sarcolemma, it then dips down into the interior of that muscle fiber. Okay? And then simply the sarcoplasm is simply the cytoplasm. Again, it's going to be a little bit specialized for what the muscles actually have to do. So for example, it'll have a lot more glycogen than regular cells will have. Glycogen can be used for ATP production. And it'll also contain a protein called myoglobin. It is similar to hemoglobin in the blood. So hemoglobin is what's going to carry oxygen in the bloodstream. Myoglobin will take up the oxygen from hemoglobin. So that's what allows preferential oxygen delivery to working muscles, is this myoglobin. All right. So I mentioned to you, and I will hide me, I mentioned to you before that the sarcomere is the basic unit of contraction, or the functional unit of a skeletal muscle. And so basically what a sarcomere is, okay, it's basically you're going from what's known as a Z-disc to a Z-disc. That distance from Z-disc to a Z-disc is one sarcomere. Okay, so anything in between. Okay. So here you have, let's see if I can get a pen to work. So here we have one Z-disc. There's another, one sarcomere. There's another sarcomere here. There's another one somewhere over here. And this would continue. There'd be many of them. So one of the microtubules that we're going to see are these actin filaments. These filaments that we'll see, not microtubules, these filaments that we will see, they're actually called actin. They extend from the Z-disc, so hopefully you can see this little blue line right here. So they're extending toward the center of the sarcomere. Then we have in red on this image, we have what are known as thick filaments. These are located in the center of the sarcomere. Again, those are in red. And all this is saying is that in this portion of the cell, there is going to be some amount of overlap between the two. Let me just kind of bring me back for a second. So basically what you're going to end up seeing, and we're going to talk about, is that here we have our actin, and basically it is going to be sort of just a simple filament, protein strands. And then this myosin is going to have sort of these little heads on it. And since there's overlap, when the conditions are correct, these heads will basically attach to the actin and move it. So it does one of these sorts of things. Okay? And as it does that, right, the ends those Z-discs come closer together, and that's how you get contraction. And again, you're going to have millions and millions and millions, probably even billions of these. And each one is going to have very little force, but billions of these working together produces a very large force. So just different parts to a sarcomere. These are just sort of these areas or regions that scientists have delineated. You may hear about these later on, so I'm just going to go over these with you. An A band is simply where it's the full length of the thick filament. It's called an A band. Okay? And so obviously, since there's overlap between the thick and thin filaments, it will include some part, that end part of that thin filament, the actin filament. This H zone is that central portion of the A band where the thin filaments do not extend. There is something called, you can see sort of black dashes, that's known as the M line. That is the center of the H zone. We'll talk a little bit more about that in a bit. But basically what's going on is that it's essentially supporting the thick filaments in the middle. So like any kind of structural structure, that's not a very good way to term it, basically, like any structure, it's sort of if it spans a large gap, it'll kind of want a bow in the middle, right? Sort of a structural weak point. So that M line is going to help support that. And then this I band would be the light band that you would see underneath the microscope. So that's basically a region that only has thin filaments, so it looks light underneath the microscope. So basically, the A band is the dark that you see, light, dark, light, dark, light. We're going to see there's basically 3 different kinds of proteins in one of these myofibrils. Okay. We've already mentioned the myosin and actin, and as I was mentioning to you, these are going to be important in muscle contractions. There will also be 2 other ones that we're going to talk quite a great deal about, troponin and tropomyosin. These are going to be located on the actin. Okay, so troponin and tropomyosin are located on the actin, and these regulate the contraction. So it will regulate when the contraction basically turns on and off. Okay, and we'll talk about how that happens. And so basically, contractile proteins and regulatory proteins are going to work together. The structural proteins, those are basically just doing that. They're providing structure to that sarcomere. Okay, so it'll help with things like the alignment of the actin and the myosin, the elasticity, and the extensibility of the muscle. And so we'll talk briefly about these other protein types. Okay. There's a video here. I would recommend that you guys maybe pause this video, go ahead and copy this link, take a look at it. It's going to give you a little bit of foreshadowing, or I guess it kind of puts together everything that we're going to talk about into a video. So it might be helpful to watch it once, get kind of an idea of how everything moves, and then come back to the video, to this lecture, and then listen to how I take you through it. Okay, so feel free to pause this, take a look at the YouTube, come back. All right, so let's kind of break this down. So again, skeletal muscle contractions is really a molecular phenomenon. This is happening at a tiny, tiny level. I know it's very hard, I think, for us to everyday conceptualize this, because muscles, I mean, they're such big actions, right? I mean, they're these big structures. When we eat hamburgers, we eat steaks all the time, so we see them all the time. And they provide so much force, so it's hard to imagine that we're getting all this force out of these tiny little molecules, but we do. Okay. And as I was mentioning to you guys, so I'm going to kind of bring you back for a second. As I was mentioning, what we're going to really talk about, so here we have our actin, and here we have our myosin. And basically, you can see in this image down here, this myosin, it has something, these club-like heads is what they call it. Okay, so but basically you can kind of imagine it kind of like that. What we're going to talk about is under what conditions is that myosin head going to be able to attach, move, detach, move forward, attach, move, detach, go back. Does that make sense? So you're going to constantly have that attach, move, detach, get back to where it started, attach, move, detach, go back, attach. So it's kind of very mechanical like that, right? So that's what's going to happen, and is that over and over again. And mind you, kind of like this image is showing on this bottom left, there's many of them. We talk about it just for the sake of simplicity, and I can't have so much going on at one time. Usually I talk about one of these heads at one time, so you'll see it's more like that. Okay, that we're going to talk about. However, remember, this is a 3D structure, so these things are really sticking out all over the place, and really it's more of like an action like this, where they're around that actin sort of cylinder, and they're really bringing things up like that. Maybe it's a little bit more accurate visually, like if any of you guys have ever worn pantyhose, and you know how you have to kind of like work the pantyhose, it's like all your fingers kind of like up your leg, it's kind of more like that. Okay? So anyway, with that, hopefully that visual, I'll kind of disappear again here so I can draw more on the screen. So here we have our actin molecules. They sort of look like little beads in this picture. On each one of these actin molecules, you have a binding site. So this sort of black, I don't know, hole or whatever you want to call that, is going to be a site to which this myosin head is going to be able to attach. Okay, and only there. It can't attach anywhere else. Right? But remember we had these regulatory proteins, this tropomyosin and the troponin. Okay, so you can kind of see these actin molecules arrange themselves in this what's known as an actin helix. So they kind of twist around one another. It's a double-stranded sort of structure. Now you have this tropomyosin is also going to wrap around that, and if you notice, it's sitting right over those binding sites. Okay, so it's blocking them. Okay, so under certain conditions, we'll talk about those, these myosin heads cannot bind to those actin molecules because those binding sites are being covered by this tropomyosin, right? And what we're going to see, you see how this troponin, these little, I don't know, bubble balls, these yellow things, are sitting on the tropomyosin. What we're going to see in a second is that those will move, and as they move, they pull the tropomyosin off of the binding site, leaving it exposed. And as soon as that happens, then these myosin heads can attach. I often think of that movement of troponin, kind of like when you're sleeping and you got the blankets and you roll over and you pull the blanket with you and you uncover the person next to you. It's kind of like that. Okay? So let's take a little bit more of a specific look at this. That's just what I mentioned. So troponin will have 3 different binding sites. One obviously is binding to tropomyosin, that's how it sticks to it. Okay. One binds to actin, and then one of them will bind to calcium, because as it moves, it'll bind to actin. And then one of these will bind to calcium. So what we're going to see in like a second, as soon as calcium comes in, it'll bind to troponin, it'll make it change its shape, it kind of rolls over, and it pulls the tropomyosin with it. There it is. So let's put all of this together. So here we have our muscle cell, right? So here's our sarcomere. So here's our little myosin head. Here's the actin and the red balls. It has these binding sites, they're kind of orange. However, that tropomyosin is covering them. And then here's the troponin, those little yellow pearls, okay? One of the things you will notice, so all muscle contractions require ATP. And one of the things you'll hopefully notice is that these myosin heads have ATP in the form of ADP plus a phosphate. Okay? So these myosin heads have what's known as an ATPase. ATPase, let me write that down for you. ATPase. Okay? And that will convert ATP, adenosine triphosphate, so it converts that into adenosine diphosphate, so 2 of them, plus P. And in this case, very similar to what we saw earlier in the semester when we talked about the sodium-potassium pump, the presence of that phosphate, okay, will either will cause a conformational change to occur, so it will cause a change of shape to occur. So when it's in this configuration, when we have ADP plus P, this myosin head is going to be cocked, it'll be bent back, ready to spring forward as soon as these binding sites are made available to it. Okay? So we consider this in the energized state. Okay? So right now this myosin head is in the energized state, it's ready to go, however the binding site is being covered. In this case, the sarcoplasm, the calcium levels are low. Okay. So an action potential, so that's what AP here stands for, an action potential from a motor neuron. It's going to go across that sarcolemma, it will go down those transverse tubules, and it will trigger the release of calcium from the sarcoplasmic reticulum. Okay, so the sarcoplasmic reticulum empties all this calcium. That calcium makes its way and it binds to the troponin. Okay? And that binding to troponin is what will cause that troponin to undergo a conformational change. Let me get my pen here. Okay. So it undergoes a conformational change. It means it changes its shape. Okay. So troponin changes its shape, it pulls on the tropomyosin, and now you'll notice these binding sites are available. Right? So that myosin head binds to it. Okay? Like I said, if it's available, it will bind to it. Boom. Now, what you see here, and is super important, is again, things are going to change their shape whether or not you have this phosphate. When it bound to that actin binding site, that basically is going to trigger the release of this ADP and phosphate. As soon as these are released, that head will move. So the ADP and phosphate separate from the myosin head. The myosin head moves, basically pulling on the actin. Okay. Now, it's stuck there. Okay. What you have to have happen is you have to have another ATP come in. Okay. As soon as that ATP binds to that myosin head, it will detach from the actin. Okay. When it's attached, the other term we call that structure of it, when it's attached to actin, we call that a cross bridge. Okay. But as that myosin is attached to that actin site, you have to have an ATP come in to allow it to detach. So as you can see here, it detached ATP. Now, look at what happens. Again, that ATPase is going to hydrolyze or break down that ATP into ADP and phosphate. And now we're back to that very first image that we saw. So now it's back in this position. So it's said to be, it recocks. It goes back to that position. If these calcium levels remain high, it'll undergo that exact same process that we had just talked about. And as long as those nerve impulses continue, saying like, hey, keep that muscle contracted because we're trying to, I don't know, hold up this couch so our friend can get the thing underneath it, they'll keep contracting. Okay. However, if that nerve impulse stops, the calcium levels are going to drop. They're actively pumped back into that sarcoplasmic reticulum, and the troponin no longer will bind to them, so it relaxes, it goes back to that state that it was before. The tropomyosin slips back over the actin binding site, and now these myosin heads can no longer bind to it again. Okay. So we call that a ratcheting kind of action. Okay, so let's just kind of take a look at it. We sort of started right here, and here you can see there's many of these, right? So it's all happening. They're kind of, again, like they're kind of walking. Sort of like how you're, not all you, like for you to walk, one foot, then the other foot, then the other foot, then the other foot, same kind of idea. So here we have, it's ready to go. We haven't had a muscle signal yet. We have ADP and phosphate, but those binding sites are, they're covered. Calcium comes into the cell. It binds to troponin. It causes troponin to undergo a conformational change, pulling on the tropomyosin, exposing the actin binding site. Okay. That allows the myosin head to bind. That binding causes ADP and P to be released. That causes the head to move, and so the actin ratchets. Oops. The actin ratchets. Another ATP molecule comes in. Just the presence of that ATP molecule causes that myosin head to detach. Okay. And then basically that ATPase in the head of the myosin will break that ATP down into ADP and P, and it goes back to that position that we saw originally. And as long as calcium levels are high and those actin binding sites are uncovered, we will undergo this circle. Okay. So then, let's actually get to that next picture. What causes rigor mortis? So if you're looking at these two fish, right, you can see this one is all stiff, this one is flaccid. So this one is undergoing rigor mortis. So I really want you guys to think about this. What we just talked about, how muscles ratchet. Something in there is going to kind of go a little bit awry. Not awry, but something in that will cause rigor mortis. I want you to think about it. And if you have any questions or you want to get confirmation on that, you can email me. All right? We'll stop there.